Have you ever stopped to think about what's happening inside your body right this second? I mean, really think about it. Millions and millions of chemical reactions are firing off inside every single one of your cells. For you to just sit here and listen, these reactions have to happen at an absolutely blistering pace and with perfect precision. So how does that even work? The answer is a group of incredible molecules that are the true heroes of biology, enzymes. In this explainer, we're going to pull back the curtain on these amazing cellular catalysts.
All right, so here's the game plan. We're going to start with the absolute basics of what enzymes are. Then we'll check out their toolkit, the little helpers they need to get the job done. We'll see how they find and grab their targets, and then we'll get into the science of their speed. After that, we'll cover how these powerful machines are kept under control, and finally, we'll connect all of this back to what it means for our own health.
So, let's dive right in. What exactly is an enzyme, and what makes it such a biological superstar? Okay, at its heart, an enzyme is a biological catalyst, and it's almost always a protein. Its one and only job is to take a chemical reaction and make it go faster, like way, way faster. Now, this is a really crucial point. Enzymes are not magicians. They can't make impossible reactions happen. They only work on reactions that are already going to happen anyway, ones that are energetically favorable. They just take a reaction that might take 1,000 years on its own and make it happen in a millisecond.
And if you compare them to the inorganic catalysts you might have used in a chemistry lab, well, there's just no contest. Enzymes are in a whole other league. They work in the cozy, mild conditions of our cells, not in some high-pressure, boiling hot flask. They are ridiculously specific. They can pick their one target molecule out of a soup of thousands. But maybe the most important difference is that they can be regulated. The cell can turn them on and off like a light switch, which is absolutely essential for life.
So how can one type of molecule do so many different jobs? Well, the secret is that many enzymes don't work alone. They're kind of like master craftspeople, and every master needs a good set of tools. Let's take a look at an enzyme's toolkit.
Sometimes, all an enzyme needs is a simple inorganic ion, like iron or magnesium. We call that a cofactor. Other times, it needs a more complex organic helper molecule. If that helper just pops in and out as needed, we call it a coenzyme. But if it's bolted on permanently, it's called a prosthetic group. And here's the direct connection to you. A huge number of these coenzymes are made from the vitamins you get in your food. That's why a balanced diet is so critical. You're literally supplying your enzymes with the tools they need to keep you running.
Now, with millions of enzymes out there, you might think it's total chaos, but scientists have neatly sorted them into 6 major classes based on the kind of chemistry they do. You've got oxidoreductases, which handle electron transfers. You have transferases, which are like a molecular moving company, shifting functional groups around. And then you have ligases, which stitch molecules together. This system basically gives us a complete catalog of every chemical job that happens inside a cell.
All right, this is where the real magic happens. How does an enzyme find its one specific target, what we call a substrate, in a crowded cell and get to work? It all comes down to a special little pocket on the enzyme surface.
For a long time, the thinking was based on a lock and key model. The idea was that the enzyme's active site was this rigid shape, a perfect lock for its specific substrate key. But now we have a more dynamic and accurate picture called the induced fit model. This model says the active site is actually flexible. It's only after the substrate binds that the enzyme changes shape to create that perfect snug fit. It's less like a lock and key and more like a glove molding to a hand.
A fantastic example of this is an enzyme called hexokinase. Its job is the very first step in getting energy from sugar. So, when a glucose molecule and an ATP molecule wander into its active site, the enzyme literally clamps down on them. This big conformational change does 2 things. It creates the perfect chemical environment for the reaction, and it kicks out any nearby water molecules that might mess things up. It's just brilliant engineering.
Okay, so here is the big secret. The real key to understanding enzyme power. Imagine you want to break a metal stick. The hardest part isn't holding the stick, it's bending it to that point where it's just about to snap. That strained, high-energy point is called the transition state. An enzyme's active site isn't shaped to fit the straight stick. It's shaped to fit the bent stick. By grabbing onto and stabilizing that unstable transition state, the enzyme makes it ridiculously easy to get over the energy hump and break the stick. That's it. That's the secret sauce.
Speaking of speed, we can't just say enzymes are fast. We're scientists. We need to put numbers on it. So how do we actually measure and describe just how fast these little machines can go?
The classic model that every biochemistry student learns is called Michaelis-Menten kinetics. It gives us this beautiful curve that shows how the reaction speed changes as you add more and more substrate. And from this model, we get two super important numbers. The first is Vmax. Think of this as the enzyme's absolute top speed, pedal to the metal, going as fast as it possibly can because it's totally swamped with substrate. The second number is Km, the Michaelis constant, and this one tells us a little more about the enzyme's personality.
So what does Km tell us? Essentially, it's a measure of how sticky or how high the affinity an enzyme has for its substrate. But here's the slightly tricky part. It's an inverse relationship. A really low Km means the enzyme is super sticky. It binds its substrate very tightly and gets up to half speed with just a tiny amount of substrate around. A high Km means the enzyme is less sticky, has a lower affinity, and needs a whole lot of substrate before it really gets going.
Now, that Michaelis-Menten curve looks nice, but if you're a scientist in a lab trying to get precise numbers from your data, it's actually pretty hard to pinpoint that Vmax. So they came up with a clever mathematical trick called the Lineweaver-Burk plot. By plotting the inverse of the velocity against the inverse of the substrate concentration, that swooping curve magically turns into a straight line. And this is so useful because it's much easier to find the exact intercepts of a straight line, which lets you calculate Vmax and Km with much greater accuracy.
Okay, these enzymes are incredibly powerful, but you can't just have them running at full blast all the time. That would lead to total chaos inside the cell. A cell needs to have control. So let's look at the clever ways cells put the brakes on or hit the gas.
A major strategy is reversible inhibition, and it comes in a few different flavors. Competitive inhibition is the easiest to picture. An inhibitor molecule that looks like the substrate literally sits in the active site, blocking the real substrate from getting in. Then you have uncompetitive inhibition, which is a bit sneakier. The inhibitor waits for the substrate to bind, and then it latches on, locking the whole complex down. And finally, a mixed inhibitor doesn't really care. It can bind whether the active site is full or empty. Each one messes with the enzyme's kinetics in a very specific, predictable way. But maybe the most elegant method of all is allosteric regulation. This is like a remote control for the enzyme. Modulator molecules bind to a completely separate spot, far from the active site. These enzymes can flip between a low activity, tense, or T state, and a high activity, relaxed, or R state. An activator will bind and lock the enzyme in that on, R state. An inhibitor does the opposite, locking it in the off, T state. It's an incredibly sophisticated way to fine-tune activity.
And the cell has a couple more tricks up its sleeve. It can stick a chemical group, like a phosphate, onto an enzyme to instantly flip it on or off. And for really dangerous enzymes, like the ones that digest your food, the cell builds them with a safety lock on. These inactive forms are called zymogens, and they only get activated when a piece of them is snipped off, a permanent, one-way activation that happens only when and where they are needed. Okay, so we've been pretty deep in the biochemical weeds. Let's pull back up and see why all of this stuff really matters for our actual, everyday health and for modern medicine.
This is just a beautiful example of biological design. Your muscles and your liver both need to process glucose, but they have slightly different versions of the enzyme, called isoenzymes. In your muscles, hexokinase has a super low Km, meaning it has a crazy high affinity for glucose. It will snatch up every last bit of sugar for energy, even when levels are low. But in your liver, glucokinase has a high Km. It has a low affinity, so it only really kicks into gear after you've had a big meal and your blood sugar is high, telling the liver it's time to start storing that excess sugar away. Same reaction, different properties, perfectly tuned for the job at hand.
This idea of different tissues having their own special enzyme versions is also incredibly powerful for doctors. It's like molecular detective work. Take an enzyme called lactate dehydrogenase, or LDH. Your heart has one version, LDH1, and your liver and muscles have a different one, LDH5. If a tissue gets damaged, say during a heart attack, its cells burst open and their specific enzymes leak out into the blood. So by measuring the levels of these different isoenzymes, doctors can get a huge clue about which organ in the body is in trouble.
So as you can see, from powering every single action that defines being alive to serving as critical clues for diagnosing disease, enzymes are without a doubt the master catalysts of the cell. Our understanding of how they work has already changed medicine forever. So the real question becomes, as our ability to measure, understand, and even engineer these amazing machines gets better and better, what's the next frontier in using them to keep us healthy?